A handful of decades after Mendel, a scientist by the name of Punnett developed a way for trying to visualize the probable outcomes of progeny for the different hybridizations. We now refer to this as Punnett square. We touched on it a couple of lectures ago, and we're going to talk once again, just kind of brush up on how this works. You'll see that for every gene that you're going to be discussing, here our gene is P. Okay? For gene P, our organisms that we're hybridizing, that we're mating, has diploid. So it's going to have two copies, two P's. Okay? Turns out that the P's come in two traits or two alleles. Big P, little p. Our two organisms that we're going to breed or hybridize are their genotypes. Remember, genotypes is what genes are present. Big P, crossing with little p, little p. Since we're only dealing with one gene, means that the gametes have only two possible outcomes. Since there's two alleles, there's only two outcomes. So it's 2 across the top, 2 down the sides. When you go to fill in, you fill in the columns, big P, big P, big P, big P, and then you fill in the rows. And then you can go through and you can look at the potential offspring. Here are the probable offspring. Since we're crossing true breeding big P with true breeding little p, all of the offspring will have a big P and will have a little p. Now, here's a word haven't used yet. Homozygote. There's homozygote, heterozygote. Homozygous, heterozygous. Homozygote. Homo means same, self. Okay? Homozygote means the same zygotes. The same gametes. Means all the gametes are going to have the same allele. Heterozygous means different. Different zygotes, different gametes. It means that all the gametes that will be presented or that will be made can have one or two possible outcomes. Big P or little p. So, the parental generation, true breeding, homozygous big P, true breeding, homozygous little p, gives an F1 generation that are all big P, little p. You go through, now you want to do the same cross again. Big P, little p. Big P, little p, big P, little p. Heterozygous, heterozygous. Since they're heterozygous, that means they're no longer true breeding. Why? Well, because look, we fill in, that should be a big P, not a little p, big P. So, big P, big P, big P, little p, big P, little p, little p, little p. Offspring. going to be different. Here's the problem. If you don't know if you're dealing with homozygous or heterozygous, here, and you look above my head, this example from the book. The only time you know, based on the phenotype, what the genotype could be, is if it's recessive. Okay, white flowered. Here it's the green pea. 2B white, 2B green, it has to be homozygous recessive. Little p, little p, little y, little y, little r, little r. Rule of thumb, in case you haven't figured it out yet, one of the tricks geneticists use when they're talking dominant and recessive genes, dominant recessive alleles, capital goes for the dominant, lowercase goes for the recessive. So little y means it's homozygous recessive. The yellow, well, since that's the dominant phenotype, only has to have at least one allele be dominant for it to be yellow. So how do you know? Well, Mendel took this into account when he was designing his experiments, and he developed what is referred to as the test cross. Given a dominant phenotype that you do not know what the genotype is, you run an experiment. You run an experiment and you figure it out. If our yellow pea is homozygous big Y, what you're going to end up with is big Y, and all of the offspring, when you do a Punnett square, will be yellow. If our unknown is big Y, little y, well, big Y is only going to occur in 2 of the possible outcomes, and it'll be little y in the remaining 2. So it'll be 50-50, yellow, green. So test crossed is you just do the experiment. Here's an unknown genotype, known yellow phenotype, crossed with a known green phenotype genotype. Okay, so you know the green, you know exactly what it is because it can only be this one thing. You cross them, you hybridize them, and then you see the outcome. Test cross. When you look at the Punnett squares, if you're following one trait, a monohybrid cross, mono meaning singular, singular hybridization, you're looking at only one thing. The possible outcomes you can do in a 2x2 Punnett square. Okay, and you look at the phenotypes of this outcome. 3 yellows to 1 green. 3 to 1. That's what Mendel pretty much saw. Remember the first slide, the last lecture, 3.14 to 1, 2.96 to 1, 3.15 to 1, 3.01, 01 to 1, so on. But you have to keep in mind, just because you know the phenotype, you don't always know the genotype just by looking at it, because the dominant trait, the dominant phenotype, could be either homozygous or heterozygous. The only time you know is when it's the homozygous recessive. So, doing a test cross allows us, and allowed Mendel, because he's the one that developed it, allowed Mendel to determine what the genotype or what traits are being carried. What two versions are present. So by going through, he could determine this known phenotype, unknown genotype. By going through and doing the crosses, he could either look at visually and determine homozygous or heterozygous. Do it for pea color, do it for flower color. This purple flower can either be big P, or big P, little p. We know since it's white, only one possible genotype gives you white. So you do the cross and you look at the offspring. If it's all purple, that means it was big P, homozygous. If it's 50-50, then it was heterozygous. That's the beauty of a test